Good morning.
Compressor is in the water. Please stand by to test the remote. Please test the remote at this time. Pay in and then pay out.
Good morning from the RV Falcor. The time is 2 minutes past 4 in the morning, local time, which is East Coast US time.
Please pay out.
It is Wednesday, June the 26th, and we have just launched the...
Everything looks good here. Confirm everything good on your end.
We've just...
Green across the board.
Roger that. All clear for Nereus.
Okay, we have just launched the data interface link that we use to connect Nereus to the deep ocean, otherwise known as the depressor. And we are getting ready to launch the Nereus vehicle. This is going to be Nereus Dive 060, and we are diving today at the Damm hydrothermal field, 2,300 meters of water, and today is hopefully going to be a long and extensive dive. We're going to see just about all the features we know from the Damm hydrothermal field, starting at the southernmost tip in the tube-worm-rich areas, working our way around the east side of the flank, visiting multiple low-temperature hydrothermal chimney sites, and then getting progressively hotter as we spiral our way inwards to the very top of the spire by the end of the dive. We should be on the seafloor by 6 AM local, and we expect about 10 hours available time on the bottom, so we'll see how long our batteries last and exactly how much we get to show you. But we're going to start with our highest priority for the day, which is doing some low temperature work down near the tube worm fields to the south of the main Damm spire. We're just showing you the view now from Nereus's cameras as it begins to lift up out of the cradle. It's been swung out. You can see Andy Bowen and Mario Gomez from Woods Hole in the picture there. Andy on the left, Mario on the right. That's Kirill, a member of the ship's crew, wandering past to the back there. So Mario and Andy have two particularly important lines. Mario is maintaining the line that is keeping the front of Nereus secure. Andy is looking after the slip pin. Basically at the moment, Nereus is suspended from the crane underneath the ship's A-frame. And there's a little metal pin on a rope that when Andy pulls on that, once Nereus is in the water, that will disconnect the vehicle from the ship completely, and Nereus will be free to begin its descent down to, in today's dive, 2,300 meters, which should take about an hour and a half or so to get to the seafloor. Oh, there is something missing from Nereus, so we're just having to return to the cradle and then continue with the launch. Okay, good news. It was a relatively minor fix, it seems. We are ready to start continuing with our deployment. We didn't have to bring Nereus all the way back. So you can see Kirill, Andy, Mario, and at the back, one of the heroes of the cruise, Miro, our chief engineer. Here's the cabin next to me. in the yellow hard hat at the back of the deck there. Keeping out of harm's way, but ready to leap into action if there are any problems with anything on the ship's engineering side. So we're pretty much at the apex of Nereus is being swung out over the side. The A-frame is lifted up to its highest point. Looking down there between the red rails on either side of the metal cradle that Nereus rests on when it's on deck. And you'll start lowering down now, coming back closer to the height of the deck of the ship. Just coming into view now is the CTD rosette frame on the left. That's the other major piece of equipment we've been using throughout this cruise, throughout first leg and second leg, collecting water samples from the deep ocean. On leg one, we were using that to try and help track down signals and guide Nereus in AUV mode of new sites to explore. And then as we've continued our exploration with Nereus in ROV mode, it's become Nereus's turn to actually be the guide to tell us exactly where we should lower our CTD to get the very best geochemical samples and microbiological samples we can from directly above the active hydrothermal fields down at the seabed. Here we go now then. Nereus is lowered over the back of the ship. The white painting you can see on the back of the dark green hull. The F for Falcor. And underneath that the words Georgetown, because that is the town in Grand Cayman Island, actually just 50 miles north of here, where the research vessel Falcor is registered. And here we can just see Nereus's lights now shining on the ocean surface from above. And you're about to see when we sink through the interface. There we are. We're into the water. We have permission to launch. And the pin is pulled. And away Nereus goes down to the deep ocean. And as Nereus begins to descend, what will happen immediately is that we will start paying out the winch for the deep ocean depressor, our data interface link, and that will accompany Nereus down for the first 1,000 meters or so down toward the seafloor. See bubbles streaming out of the basket today. We have a slightly different basket from yesterday, and we'll, as we get closer to the seabed, we'll be doing our standard game of playing what's in the basket today, but that'll be for a later time.
Once you get about 20 meters down, you can slow and match Nereus. Alright, it looks like you're clear, so just follow Nereus
And
at your leisure.
rather interestingly, we have about 5 or 6 different views of what's happening on the ship that we can see. At about 50 feet down, 16 meters down. Interestingly enough, if you're looking over the back of the ship, you can still see the lights on the front of Nereus as it carries on down towards the seafloor. Hello again from the RV Falcor, and we're approaching 5 o'clock in the morning, and it's time for our daily dose of separation anxiety. We are getting ready to stop our depressor at 1,000 meters, and at that point we will send the signal to release the catch of the float pack, which will separate from the rest of the depressor. The float pack and Nereus will then continue the rest of the way to the seafloor. So stand by, and we'll let you know as soon as that has happened. Earlier, today Chip has that configured so you can take just continuing our tour. We believe there was a slight internet connection, so as we said, we have the manipulator arm on the far left of our field of view, the bio box on the far right. In the middle we have the blue scoop net that we're going to use to collect any mussels. The silver cylinder with the plastic tube coming out the back is going to be used extensively today. It's both the inlet for the super sampler, where we take our microbial samples, but it also has a temperature sensor, so we're going to be using that today. Every time we find diffuse fluid flow sites, we're going to stop and check out their temperature. And then front and center at the top of your picture with a bright yellow T-handle, there's a little instrument today. It's basically art on a stick. We'll be telling you more about that later on. But we have one of two small spheres, the little silica marble planets. So, what's in the basket today? Take two. And we'll, so we're going to do from top to bottom this time. I apologize, we were doing the what's in the basket today earlier. Some of you were probably listening along and our internet connection went dead. So we're going to try that again. Just waiting for the cue from Jeremy that we're good to go. Yep, we're up and live. So, from the top of the basket, working our way down. On the left you can see our craft manipulator has 6 degrees of freedom. On the top right we have the black square that you can see is the lid of our bio box. It's thermally insulated, full of water, and if we have any biological samples or indeed any really good rock or sulfide samples, we'll be putting those in later today. We put the lid back down and we hold it in place secure over a little latch with a bungee cord. In between the two, you see a black plastic milk crate. We have three things in there. We actually have a little piece of installation art we're going to be putting on the seafloor. It's a marble planet, or made of silica glass actually, made by Josh Simpson from an arty studio in northwestern Massachusetts. We're going to be putting out on the seafloor this morning as part of his Infinity project. Then just to the left of that, you see a large silver cylinder with a plastic hose coming out the back. That's the inlet for the super sampler. I'll tell you more about that as we work our way down the basket. And then the last thing in the basket this morning in the front there is a blue nylon mesh scoop net. We're going to be using that. One of the holy grails for today is to try and find some mussels living around the vent site. We've seen lots of shells of mussels at this vent site in our past dives. We've also found mussels of very similar nature at other seep and vent sites, both in the Gulf of Mexico and along the Atlantic margin, and also out at the Mid-Atlantic ridge. So we'd like to know what these mussels are most closely related to. Are they more related to the mussels at the ridge site, which seems to be the case for the shrimp we've been seeing on our dives here, or are they more related to the Gulf of Mexico cold seep sites, which seems to be the case for the tube worms that we'll also be seeing later today? Okay, Chris, if you could pan our camera down, please. And in the midsection, what you see on the right in another one of our black plastic milk crates are the two IGT, isobaric gas-tight fluid samplers, that were sampled yesterday. Routinely. That these things can recover fluids from the seafloor at in-situ pressures, so that's really important because it makes sure none of the gases that are in the seawater down at 2,000 or 5,000 meters, depending on which site we're venting at, bubble out as they come up to the surface and come out of solution. By keeping the fluid at in-situ pressures down at the seafloor all the way back to the laboratory, Jeff and his team can then extract those gases and get the true concentrations rather than some lower concentration, which is what gets measured in just about any other way that you sample water samples from the bottom of the ocean and bring them back to the surface. And then just to the left of that, you can see a long corrugated hose snaking out and around the basket, around the back of the post that the manipulator arm is mounted on. The T-handle there is actually where we pick that up. That's the front end of that snorkel is actually held there. We pick that up with the manipulator arm by that little silver bar that's running horizontally just above the snorkel. And that's what we use to actually slurp animals up from the seafloor. We'll be using that to collect shrimp later today, and that's another way we might try and collect mussel samples. And then panning all the way down to the very back of the basket, furthest inboard, our camera's pointing almost vertically straight down now onto the back of the basket. What we see here, you can see that same corrugated hose disappears off the bottom of your screen. That's actually going into the slurp sampler. At the very back there is a large canister. We basically slurp water and pump water through out the back of that canister, almost like a vacuum cleaner. And that's basically the way the shrimp go to when we're actually collecting them. What you can also see there on the left, you can see two rectangular objects, one solid white plastic, the other one has like a brown lid to it. That's basically the solid white part is the pumping mechanism for Chip Breyer's super sampler. We'll be using that for collecting microbial samples today. And then the bottom left is actually where all the electronics are in an oil-filled canister. That's basically how we take complex electronics down to the seafloor without letting any of the seawater get in. It's in a pressure-balanced, oil-filled compartment there. And on the right-hand side, you can see this rack with a row of 6 samplers down one side, one on the right-hand side, and then empty slots where additional samplers can go. This is where the samples actually collected from the super sampler, which is the other inlet we had in the forward basket. A couple of days ago when we were doing work at the Picard site, we were rising directly above the BB vents, the hottest vents we know in this region, and we were doing that to collect geochemical samples for Chip at different heights above the plume. Today we're using it in its second configuration as a microbial sampler for Julie Huber and Julie Revaillot from the Marine Biological Laboratory, a sister organization also in Woods Hole in Massachusetts. And they're going to be using that sampler to go and collect microbial samples from various sites of low temperature diffuse flow in and around the southern and eastern components of the Damm hydrothermal field. So that completes our tour of the basket. Thank you very much, Chris. And we are now at a depth of 1,800 meters. We have about 500 meters to go till we arrive on the seafloor. We're descending at about 25 meters a minute. So we should be ready to drop our descent weights and start settling out in about 20 minutes from now. We'll then have a little bit of driving to go. So we should be actually in place on the seabed. Given that it's 5:30 now, East Coast time, we should be settling out on the seabed sometime around 6 a.m. local time. And if you're diligent enough to be watching in California, that'll be about 3 a.m. So bless you for being tuned in right now. Or if you're watching this on the recorded YouTube channel sometime in the future, where were you? Why weren't you here for it live? And finally, if you're watching in Hawaii, it must be somewhere around midnight or 1 a.m. And I just gave the secret away. You don't have to carry on watching live because you can watch this anytime you like on YouTube. Good morning again, everybody, from the RV Falcon. It is now 5:30. We are about to take the What's in the Basket today, take 3. And if it doesn't agree in text with what you heard before, the trouble is we've just been alerted by Victor Zhukov, who is paying attention somewhere in the world, California, I believe, who pointed out that we were showing you our navigation screen instead of the picture of the basket. So what we've just done for a bit of fun is we've just had Chris move the manipulator around, move everything around in the basket, because we didn't want you to get bored and hear the same words twice. And that's also my caveat for proving that I'm not just making this up as I go along. We'll see if I actually describe the same things in the basket now as I did about 10 minutes ago when we wouldn't let you actually see what was there. Or in fact, if we were really cute, if we had Julie Huber online right now, what we would do is have a go of Kim's game, and you from memory could tell me what we would expect to see as we pan down through the basket, and you could send it in by Twitter. But we're not going to get Julie out just for that purpose. So instead, this is our third attempt to tell you what's in the basket today. So, top left we have the craft manipulator, which has 6 degrees of freedom. Don't let anyone tell you it's 7. Which can actually move around. It basically has a shoulder joint, an elbow joint, a wrist, and it can open and close its claws. That's on the left hand side. In the middle, in the milk crate, you can see the silver cylinder with the plastic tube coming out the back is the sample inlet for the super sampler. We're going to be using that to collect microbial samples today, but also to probe for temperature conditions in any of the multiple different sites of fluid flow we expect to encounter as we work our way up from the south around to the east to the northern end of the Damm hydrothermal field. That's the general plan of the dive, ending up working all our way around, spiraling in to end up right at the central spire at the summit of the Damm mound. Next to that, we actually have a little yellow T-handle. You can see there's a small silver cylinder you can see with rubber bands all over it. We'll be telling you about that later on. That's a little piece of artwork we're actually going to be depositing at the seafloor at the first site we're going to today, a particularly picturesque site, and we'll be talking about that more when the dive begins. And then in the bottom of that black plastic milk crate is a blue nylon mesh bag that we'll be using for scooping mussels if we find them, which we very much hope to do today. And then on the far right with the black lid, that's our bio box. It's an insulated plastic box with a lid that we actually can release and then close, held in place with a bungee cord. And if we collect any good biological samples, that's where we'll put them to make sure they don't get washed out of our basket when we get back to the sea surface for our science team. Panning down, we have Chris Taylor, our pilot, on the controls today for the camera. What we see here, snaking around the basket on the left-hand side, that long corrugated hose is the inlet for our slurp sampler. That's the other way we're going to be collecting large biological samples today, and we're going to be slurping for shrimp from different locations around the low temperature parts of the hydrothermal mound today. And on the right are two of the IGT or isobaric gas tight samplers that we use, or that's Jeff Seaward's team. Jeff and our jointly supervised grad student Jill McDermott use those samplers to collect vent fluids. We are very pleased to report we brought 6 of those samplers to sea with us on this cruise, and we're going to be taking 7 home, because yesterday we managed to pick one of these fluid samplers up that we had to inadvertently leave behind on the seafloor 4 years ago. We had a minor crisis on the seafloor with our ballasting. We managed to redeem ourselves yesterday by tracking down that 4th sampler. It had been left about 500 meters west of where we were diving on the vent site. We drove off into the darkness and managed to relocate that, find the sampler and bring it home. So we're going to be using those to collect fluid samples today. Continuing down, what we see at the very rear of the basket, coming into view now, is the super sampler. Remember, that's the inlet at the very front of the basket we're going to be using for probing for temperature signals all over the Damm mound today. The left-hand side shows where the pump is, that's the solid white block, and where the electronics are in that white rectangular component that has the brown center to it. That's where the electronics are for the pump, and on the right-hand side are where all the samples get collected. Today we're in microbial mode, so that's where we're going to be collecting samples for Julie Huber. And finally, the corrugated hose disappearing off the bottom center of the picture is heading into our slurp sampler that's just been purpose-built for this cruise. That's for collecting our shrimp samples. They basically go into a large Perspex canister at the back of the vehicle, and that's hopefully where between 50 and 100 shrimp will be going within the next few hours. And that completes our tour. We're hoping that's third time lucky, and you both got audio and video and internet connection without disruption. And we are now at 2,040 meters, getting very close to the seafloor. We should be dropping our weights in the next 10 to 15 minutes, and we should be arriving at the seabed sometime shortly after 6 o'clock East Coast time. So a quick update again from the RV Falcon. We have now settled out, dropped our descent weights, adjusted our ballast, and the vehicle is sitting quite happily about 100 meters above the seafloor and about 150 meters west-northwest of where we're going to be heading to. Let me just see if I can actually show you some of that on the navigation screen. In fact, I'm not going to pretend that I don't know what I'm doing. Let's see if I can switch channel to not that one. There we are. You can now see on your screens and navigation screen. What you can see is at the center is Nereus's current position. The little yellow grid lines on the squares you can see are about 100 meters on our side. Lines of longitude going north and south, up and down the screen. Lines of latitude going east and west across the screen. And you can see a whole series of little blue crosses off to the right, which are going to be different places we want to go and visit over the course of the day. We're going to be starting down at the very bottom, off to the bottom right. The top right hand corner you can see a series of concentric red circles. That's actually the main central cone of the Damm hydrothermal field. We intend to end up right at the very summit of that, at the highest temperature black smokers at the end of our dive later today. But to start with, we're heading down pretty much due southeast. You can see on the top of the screen, there we can just see that we're going to southeast about 350 meters to where we left one of our markers in 2012. And what we're going to be doing there, starting off, is going to a site that is quite unique in terms of vent biology anywhere on the planet. It's a place where we have hydrothermal tube worms and hydrothermal shrimp living, coexisting side by side at the same chemosynthetic environment where hot chemical fluids, chemically enriched fluids, are coming out of the seafloor. And we have both shrimp with their chemosynthetic bacteria and tube worms with their chemosynthetic bacteria, converting chemical energy from the interactions between the geology and the seawater to release chemical energy, and then the microbes taking advantage of that chemical energy to convert that to fix carbon from carbon dioxide in the ocean to organic carbon, both for the microbes themselves and for the larger animals that host them, the tube worms and the shrimp. So that's where we're headed first, and we're going to drive through the water column in the first instance. We should then be descending slowly. You can see as the contour lines go from red through orange, through yellow, through green to blue, it's getting progressively deeper. So we're going to be starting at the deep bottom right hand end of that little spur you can see running down to the bottom right at the corner of our map. We're going to head there first, then work our way up north-northwest along that ridge, and then rather than go straight up to the top of the spire, we're going to contour around to the east, to the right hand side, circle our way around, slowly spiral our way up to a point due north of the central cone where we know there are some higher temperature vents, and then end the day, we hope, by continuing our spiral inwards and coming out to the very top, at the very pinnacle of the spire for our highest temperature vents that we know. So I will leave this navigation map up for now, and then as we get closer, we will switch back to the view from the basket once we can see the seafloor and there's something to look at. So here we are on dive time radio, transiting our way across the seafloor in the rush hour. We have no submersibles ahead of us, and the radar suggests we have a clear run to our target site. We should be there in about 20 minutes time, arriving on the seafloor probably just a little before 6:15. So as we're watching our navigation screen right now, we're just showing you the video feed for that. It gives you an idea of the orientation for the day. We are just spiraling in with Nereus. We're going to come in the little pink cross with the cursor is showing you our first target of the day. That's the first tube worm site we're headed to. So we're going to come in, approach that from the southeast, get down to the seafloor, and then drive up the ridge towards that first point and carry on in that kind of orientation through a few of those blue crosses, picking up interesting biological specimens along the way, and also studying any fluid flow sites as we get to those different locations. So as we just cycle in now, what we're actually planning to do is to bring the vehicle down gently to the seafloor, pointing northwest a little to the southeast of our primary target for the start of the dive. So what we can then do is work our way gently upslope over the seabed from the southeast, heading in the direction we plan to go for the next hour or two, I would predict, as we stop at interesting fluid flow sites along the way and taking samples as we go. So we're just switching you from our navigation feed to our high definition camera now. We are coming down from the seafloor. We've just in the last few minutes come from 75 meters to 50 meters altitude. And the next 2 or 3 minutes we should have the seafloor in sight. We're arriving at a seafloor target depth of about 2,380 meters. We're currently at 2,330 meters. We are about 100 meters southeast of the base of the cone of the Von Damm spire, and we're going to be working our way along a ridge populated with a number of diffuse flow sites, and where we've previously seen both tube worms and shrimp, indicative of chemosynthetic fluid flow, or fluid flow that can sustain chemosynthetic microbes, and the animals that host those microbes as symbionts. So that's where we are going to arrive very shortly. And here we are, we have the seafloor in sight. I'm afraid we were slightly distracted because we were discussing whether or not Casey really does know how to dance like MC Hammer. But it's too late for him to find out now, because he's about to take over the pilot's chair from Chris Taylor. Thank you, Chris, for getting us safely to the seafloor and getting us in the right place. We'll see you later, and I promise we'll save the really hot vents for after Casey's gone back to bed. So what you can see here is a view that we've become very familiar with, working with the southeast corner of the Von Damm spire. We can see boulders of rocky material, which our past experience shows us is predominantly a mineral called talc. It's basically a magnesium silicate mineral, which is consistent with one of two options. Either it's a chemical precipitate from the fluids that are actually emitted from the Von Damm mound, or alternatively it may be that these were once ultramafic rocks that were crystallized out in the mantle 6 kilometers below the seafloor, have been pulled up and exposed at the seabed along a long-lived fault, which basically has generated the entire mountainside that we're diving on. And then the hydrothermal fluid flow through those rocks has been so extensive that it's only these very last dregs of composition that are left behind after all of the other more soluble minerals have been leached out of them. So, if you bear with me a moment, I'm just going to have a few words with Casey and scope out what we're going to be doing to get the dive started. We have a holothurian in our field of view there, just above the basket. And we can see lumps of rocky material cascading down this steep slope. And what we're going to do is work our way up the sedimented slope until we get to the top of the ridge. We are currently headed just a little east of north. Once we get up onto the top of the ridge, we're on the southwest corner at the moment, or western flank. And when we get to the top of the hill, we'll be taking a left so that we're headed more northwest and headed off in that direction. What we see immediately here are the shells of mussels. These long elongate slight curvature to them are the disarticulated halves of mussels. One of our holy grail targets for today is to try and find anywhere where there's a thriving bed of those mussels so we can take some biological samples. We have our scoop net with us that we will sample the animals with, and the bio box would be our ideal target of where we will put them once we have them. So we just had our watch change that Chris Taylor has rotated out the pilot's chair for Casey Machado. James Kinsey, who's been navigator guiding us to the seafloor, has been handed over his seat to Mario. And since one of the primary targets for the first part of the dive is looking at all the different kinds of large animals that live in this area, it should be no surprise to you that the first scientist to join us in the control van this morning is Cindy Van Dover, Director of the Duke University Marine Laboratory and our expert on megafauna from hydrothermal systems. As soon as we get set up anywhere interesting, I'll hand over the headset to her and Cindy can tell you what it is we're looking at. So we're continuing upslope right now. We're at a depth of 2,372 meters, 8 meters above the seafloor. Straight underneath us, but as you can also see, the seafloor at 23,700 meters, 2,370 meters is also right in front of us as well. So we're still passing over plenty of thick sediment as we come up slope, but you can now see an increasing concentration of rocky boulder material, which tells us hopefully that we're getting close to the spine of the ridge. And that's where we're expecting to find the sites of fluid flow. Got something light off to the right. I think I see some tube worms down low there. You see those, Cindy? Tube worms down on the right on the rocks? Yeah, should we go have a look at some of them? And I think there's some shrimp straight ahead in the top of your field of view as well, that white patch. Well, let's go down, look at those nearest tube worms, kind of just in the center of your picture now, yep. So this is the first time this cruise that we've been able to show you tube worms from the Von Damm hydrothermal field. And without further ado, I think I'm going to pass over the headset and let Cindy tell you something about what it is we're going to be looking at.
The tubes penetrate down into the seafloor, into cracks where there's warm water or where there's fluids anyway that are rich in hydrogen sulfide. And once we settle in, we're going to take a peek at the head ends of these animals. I'm looking for a plume, a red plume that could be really quite beautiful when their plume is showing. I don't quite see a hint from this video of whether the plume is really there. These animals are famous for their exquisite adaptations to the deep sea environment that they live in. They have no mouth, no digestive system. When their relatives were first discovered, the giant tube worms on the East Pacific Rise and Galapagos Spreading Center in the Pacific Ocean, Eastern Pacific Ocean, there the tube worm species is about an inch in diameter and can be several feet long. Oh, there we go with a close-up zoom, and we're going to just focus a little bit. So when they were first discovered, this tube worm, this type of worm, there was a lot of interest in how they could be making a living. And we've learned since that the plume that they have, the red plume at the tip of the tube, is gill. And it takes in chemicals from the surrounding seawater, so fluid from the seep or vent fluid, and then oxygen from seawater, carbon dioxide from seawater and vent fluid, and they use those chemicals in a process that we call chemosynthesis to make cellular material. These tube worms are really interesting because they are so solitary. Where we find them in the Gulf of Mexico, they will be in big bouquets or clusters. We don't really see that here. There are denser sites in the area, so we'll be looking at some of the denser sites. But for right now, we're trying to get a close-up and personal view of this one tube worm. Now this is a bashful one. Can we focus on the tip? So it makes it really challenging. We can't tell if these are live or dead. And we're not going to sample any of these. We want to leave them here, so we won't know. Once the chemicals are taken up by the tube worm through the plume at the tip in a live animal, then the dissolved sulfide and oxygen and carbon dioxide is transported in the blood system. These guys have a really very well-developed vascular blood system. So those chemicals are transported down to a special organ called a trophosome. And the trophosome houses bacteria. So they're endosymbionts. That relationship between the host tube worm and the bacteria was first reported by Colleen Cavanaugh back in the late 70s, early 1980s. So it's a relatively new understanding of how bacteria and invertebrates can live. It's such a very dramatic, large animal to be living entirely on bacteria inside its body. Okay, we didn't see anybody willing to show up out of these tubes. Either they're dead animals or the animals are just tucked inside the tubes. We're going to move on. I'll pass you back to Chris.
So, although we didn't get the animals to come out and show themselves today, what is kind of neat is that our navigation is completely locked in and dialed in. We're on the ridge right where we wanted to be. And in fact, this is actually a little further south and east than we've seen tube worms previously on our other dives. But it was something that Cindy had actually gone through all the photographs we took from Nereus in AUV mode early in the cruise, and this was one little patch that we knew down to the southeast of where we'd done a lot of our time series work previously that looked like it had extra tube worms as well, so it's been kind of cute to come here and check that out and see where these animals are living. And what we're going to do now, we're actually facing east at the moment onto that ridge, but now we've picked up where this rocky spine is. We're basically going to follow along this linear feature where we actually have rocky boulders sticking up, rising up above the sediment, and we're going to swing around towards the northwest and head on up the spine. And the next place we expect we're going to stop, unless we find anything interesting along the way, is that we're going to come to the site, the marker X18 site, where we've previously put out colonization experiments, a bit like the ones we saw yesterday from Adrian Glover and Diva Amon from the Natural History Museum in the UK. Cindy did similar things back in 2012 when we were here. We put out some similar experiments for the duration of the cruise, and there are further experiments sitting on the seafloor that we're going to go back and hopefully check out and see how those are cooking. So now we're swinging through. We're currently pointed northeast and continuing to swing round through due north, and then we'll be heading out. So you can see more of this talc rock, different levels of sediment cover, and here we have a whole bunch more of those tube worms that we're going to pass over as we head on up. And it looks like it should be about, well, less than 100 meters in this direction is where we're going to get to the place we wanted to get to. But I'm going to let Cindy sing out anytime she sees anything interesting biologically that she'd like to take a closer look at as we work our way up. It's like some kind of white shell, Cindy, just at the top of the screen, just halfway off to the left. Could be the inside. The mussel shells, when they open up, are they white on the inside or are they brown? Okay. So Cindy, that thinks it's probably more likely to be a clam, but we did see mussel shells as we were approaching up the sediment slope. Again, just passing off to the bottom left of the screen, another one in the center of your screen right now are two more of the examples of these tube worms. And it turns out you really don't have to be a terribly sophisticated biologist or geologist to actually find your way around and find interesting things on the seafloor. The great thing about the way nature works is that the rocks on the seafloor tend to be dark, the biological animals tend to be white and translucent, so long as you don't even have to worry if you're colorblind, that if you can tell the difference between black and white or dark and light, chances are you too can actually find interesting things on the seafloor if you were sitting where I'm sitting today. So you can see some large white shells again at the very top of the screen, just a little right of center. So we're going to go nudge over and go take a look at that. I'm just eavesdropping on what Cindy's putting into the log. I think we identified that as a vesicomyid clam. But it's not a live one, Cindy, I don't think. Do you think? No. Is that... Okay, do you want to switch to the close zoom? Oh. Is it the tube worm on the left do you think is showing its plume? Potentially? Let's switch to the zoom camera and see what we can see. Are we okay to do that just here, Casey? So let's cycle the cameras, please. Look at the clam first. Sure. I think we've just arrived at the seafloor and we've got 100% battery and we've got up to 10 hours to spend on the seafloor, so I think if we can spin left to get the clam in the center, and then there was a tube worm off further left again that we think had its plume out, not that tube worm. We're just setting up the vehicle itself first, and then we can start worrying about moving the camera around. Are you leaning against a rock or something? I think the vehicle just bounced off a rock, so we went the wrong way there, but... So that looks like disarticulated halves of a clamshell, but nobody home. Right? Vesicomyid clam. So if we... And what we're going to do is take a look a little to the left in a moment and see if we can see the plume out on the tube worm that's due left of us, I believe. The great thing about this being a shallow site at Damm is that we can get down to the seafloor nice and early. We don't have to leave the seafloor in any particular hurry. You can just see top left of our screen now is a tube worm pointing in just the right direction. What we'll do is have a look over there with our camera in a short while and see whether we can't see down inside the top of the tube to see what's there. Would it help to switch between cameras, Casey, for the flyby? Let's cycle back to the wider field of view. Then when we get things centered up, we'll switch back to our zoom camera. So what you can see now, just off to left of center in your field of view, ahead of the basket, is a tube worm sticking up out of the sediment with its open end towards us. As we center up the basket, we should be able to zoom in and have a look in through the top end. So we're just doing a quick little bunny hop off to the left with the vehicle. As I was saying earlier, because this is a shallow site, we have up to 10 hours potentially to spend on the bottom today. The only reason we'd need to come back earlier than that will be if we run out of battery power. So we're going to take our time to look around. Our fastest way of burning through energy is when we're using the manipulators. So we've got plenty of opportunity now. We only have a limited amount of samples we can take. So we can take our time to have a nice look at the biology as we drive. Cindy and I are in the van enjoying the exploration and what we're going to see, and everybody else was working flat out through the night. I think Max was the last person processing samples to go to bed. He only went to bed at 5 o'clock this morning. He'd been up working through the night. So none of those guys are going to complain too much if we don't start sampling straight away. That's right. I think there's scope there. We know the plumes, certainly there's no plume extruded from that. And the question is whether or not there's anybody home that's retracted into the tube. On a good day, you can actually see right in the end of the tube, you can even tell which species it is. But they deliberately have this defense mechanism that we've seen them in other places, that other animals come along and prey on the soft tissue when these things do emerge. So it's not unusual for them when they're not feeding to basically withdraw into that tube, which is made of chitin, basically the same kind of material our fingernails are made of, is their external armoring. Can we bring it any sharper focus, Casey? Just at the very tip, the uppermost part. Can you see where is the bright white looking at the inside of the tube? It's one of the interesting things we're learning to adapt to with these new cameras we have on Nereus. They're the ones donated recently by James Cameron to Woods Hole along with the Deep Sea Challenger vehicle. So the good news is that these are high definition cameras that are rated to 11,000 meters, so anywhere Nereus can go, these cameras can go. What we do have is a fixed focus, wide angular fixed focus zoom lens. What we don't have is the luxury that you might have on your digital SLR camera at home, that you can zoom in and out anywhere in between the two. So I think we can just see, well, Cindy and I know what we're looking for, so I think we can convince ourselves we can just see the soft tissue emerging from the inside of that tube. And you can see concentric rings now as you go down from the bright white disc at the center. As you go down the outside, you can see almost like concentric rings around these things, the structure away from the core of the tube. Okay, shall we move on? Okay, let's switch back to our wide-angle camera, please. That was really good, yeah. That's a nice way to test what we can do with these cameras. And if we find some less bashful tube worms later on, we should be able to get some very nice video. So now the time aboard ship has just gone 6:30. We're at 2,370 meters depth, sitting on the seafloor. We are about to lift back up and continue our trajectory to the north and west along this rocky spine of hydrothermal mineral. We sampled this rock in numerous occasions across the study area at the Damm site. Everywhere we've been, when we pick this material up and bring it back to the lab to analyze, it's proven to be predominantly a talc mineral, a magnesium silicate consistent with chemical alteration and hydrothermal weathering of ultramafic rocks, basically rocks that shouldn't normally be expected to occur at the seafloor. We believe they occur at the Mid-Cayman Rise because it's an ultra-slow spreading ridge. There are long periods where there's no volcanic eruptions, no magma generation, and instead we have long-lived faults that basically slide one tectonic plate apart from the other and expose rocks from deep inside the Earth's interior. These were rocks that probably were originally crystallized 6 kilometers beneath the seafloor. So when you think even our deepest vent sites we've been to are already 5,000 meters deep, but then another 6,000 meters deep below that into the rocky, toward the rocky center of the Earth, it's where these rocks originally crystallized. They've been brought up to the surface, and it's their interactions, it's the unusual chemistry that can result from that makes these chemosynthetic sites, or these hydrothermal sites, particularly intriguing, because the reactions that can take place here, and we can treat them almost as a natural laboratory, coincide with some of the conditions that people think might have been prevalent at the very early stages of Earth history. As you can see from our view here, as we were heading north, we can actually see the hillside sloping down to our left. We realized we're not actually right on the spine of the ridge, so we're just going to turn into the rocky face and start climbing uphill till we actually come back up on the, we know it should be about a 5 to 10 meter wide plateau at the very top. You have a bright white squat lobster and its legs splayed on the black rock just in the top right of our screen right now. And there's something white and blobby hanging out behind a rock to the left, which we think might be a zooarcid fish. And we're continuing upside. Oh yes, that little fish has revealed itself now. And another. Morning, Julie. Did you get my message in the log?
I did.
Julie is such a technophile that the first thing she does when she wakes up in the morning is she actually checks out on her iPhone what's been happening in our event log, we have this great way of actually logging things electronically from anywhere on the ship using the ship's Wi-Fi. So I knew Julie was setting her alarm clock for 5:45, and then she was going to work out whether she was needed in the control room yet or not. So we actually, in the event log today, actually put in a "Good morning, Julie" message so that she'd know what time we were on bottom and when she needed to be here. So I'm pretty sure it won't be long till she'll be here joining in, providing her running commentary of the day, and probably tweeting simultaneously and generally interacting with social media any way she knows how. So we're just spinning back around now. You can see we have lumpy, rocky substrate right in front of us. It's downhill to both left and to right. And what we're going to do from here is we have a range and bearing of where we should go to find the next target. So Casey, I would be inclined to come back round for now and go due north. If we swing back to right, we could see along the top of that ridge. We could just follow along the ridge top rather than head out into open water. And I think it's going to curve around to the left, and it'll take us where we're headed. Generally where we want to be is a target that's actually about 30 meters away northwest of us. But rather than lose the ridge axis, let's drive forward over this sediment, get across that, and then when we see the next lumps of rock, we can start turning as we see fit, depending on what we see between here and there. You see again more white discoloration or a change between dark and light in the image directly ahead. Quite often when we see that, the light is indicative of biological activity. So again, that looks like mussel shells around the base of the rocks on the right there. And again, dead ahead as well, I think. Are those, you can see those are dead shells. What we'd really like to find are live animals today. Those are mussel shells, not clam shells, Cindy said. We can see a couple of those squat lobsters. So we're going to continue along this heading. And Cindy's keeping a keen eye out if she sees any evidence for any live animals rather than dead ones. See a few little shrimp on the top right underneath a little rock there. Let's carry on up over the top of the rocks. We have more tube worms to our left, so it's probably a good time to start bearing around to the left now, Casey, and changing our heading. Right under us there, you can see there's a little crevice, white colours in there. There's going to be shrimp in those little crevices. And we're arriving, we believe, at our marker X18 site. We see the marker just in the top field of view. In fact, that's one of our rulers. So when we put that out, you can see there, when we put that out about 18 months ago, that was a flat strip of card on the seafloor with a known length, so we could fly over it and measure how big things were in our field of view. So I think, Cindy, this might actually be that site of fluid flow there, Casey, right in front of us. That might well be the first site that Julie wanted to investigate. So let's settle up here. And what we might want to do is actually look around. There's a large marker on a yellow cord with X18 water jetted into the white plastic. And I think the whale experiment was right by the marker, wasn't it? Although Is that it, sideways on? I believe it is. We only put one down, so let's just sit here for a moment and let the dust settle, shall we? So while we're just waiting for the dust to settle, I'll paint you a picture in words of what we're going to be seeing in this location. This is somewhere that we was first imaged by the Hibis camera system that was used here on the James Cook in 2010, that people had seen the worm tubes down along this ridge. In 2011 we came back here with Paul Tyler from the UK and myself on the Okeanos Explorer, and we got close enough to these animals to actually see the live tube worms and to actually see the tube worms and shrimp coexisting at the same location. And then in 2012 when we came here again, Cindy put out some colonization experiments which we're going to go and track down on the seafloor near here. Those systems were then revisited again by one of Cindy's students on the UK cruise with the ISIS vehicle, which is the sister ROV to Jason, operated from Woods Hole. And then that was about 6 months ago. We're now back here again in 2013, and Cindy's going to be back again in August, and we'll visit that again later on. So we're just looking around on this rocky terrain here. How would you feel, Julie, about having a dig around in some of these little crevices and seeing what the temperatures are like?
We
Or would you rather?
could do that, or we could just... I can't even see the flow right now. Is it sort of between there?
I don't know, we might, maybe if we switch to the zoomed in camera and have a look there, see if we can see
Yeah.
any flow. So we're just switching between cameras. I have a sneaking suspicion that the light coloration on the underside of the rocks might coincide with some fluid flow, but the best way is to zoom in. I think where those shrimp are
Yeah.
is potentially of interest.
Yeah.
So I think in that little crevice it's running, that way right across the middle of the field.
Can you reach that?
You know what? That could actually be your little square rock. That's the square rock from 2011.
Yeah, and we took a sample there, and it was about 20 degrees.
Okay. And that was the one where you had the microbes that were more like...
No.
That was somewhere
The
else.
50 degrees was. Which I know seems weird.
So is it worth the probe with the temperature probe, or... Let's do it anyway.
Yeah.
Can't do any harm, right?
Yeah.
To have a look with the temperature. Give Casey some stick time. Our temperature... Well, we have Casey as our temperature sensor. He was pretty accurate yesterday just by looking, so let's test him before he picks up the stick. And say, hey Casey, what's the temperature in there? So, we're about to try something I've not been involved with before, which would be kind of cool here. When Cindy and I were doing some exploration about a year ago, we were doing it all through virtual reality at the Inner Space Center in Rhode Island, which is actually one of the many routes that you can actually access this Schmidt-Ocean Institute cruise online right now, we understand. That we were driving up, we were visited by an artist called Josh Simpson, came to visit for the afternoon, who runs a program called the Infinity Project. He basically handcrafts small silicate marbles, like little miniature planets, that are made of the same kind of glass, the same silicate minerals that the local seafloor is made of here. And he runs a program where if you write in and you have a cool place and an interesting place to go, you can write to him a nice letter, and if he likes your letter, he will give you one of these little marbles to go put somewhere interesting on the planet. And all you have to do is promise that you'll take a photograph of where you put it. So this square rock is very iconic. It was the first place in 2011. I was getting all excited about the fluid flow I could see around this rock, and it was somebody actually watching via telepresence in the UK. Diva M1, I think, was the first person to actually shout it in my headset, but somebody else was texting from the UK saying, hey, there are shrimp there as well as tube worms. We've never seen that anywhere on the planet before. So the same little square rock in our photo mosaics, Cindy has been able to recognize over and over again as a place we come back to. So what we're now going to see on the basket, for those of you who are here for take 1, take 2, and take 3 of our description of what's in the basket today, of which I'm the only survivor in the control room, I see, looking around me. It took 3 attempts, but what we actually have is a little yellow T-handle sticking up, and on the end of that is one of these marbles from Josh Simpson. We have 2, one to put out here at the Von Damm site, and another one to put out at the Picard hydrothermal field. And we're going to leave these here for posterity, so when people come back, it's our own, slightly different version of a marker, but which will, in time, presumably just be dissolved away, and won't he be here forever, but it'll last as long as it possibly can. And if you still don't quite understand what it is we're talking about, all will become clear once Casey picks it up out of his out of the basket. So here you can see that we have this in the T-handle. Maybe Casey can just hold it up to the camera for us to look at. In fact, so we have a little metal base, and then attached by a couple of rubber bands on the bottom is this little blue silica planet made by Josh Simpson. This is what we're about to go put out on the rock as part of our Infinity project, or one of our contributions to the Infinity project. When Josh came to visit, he explained to us that he'd actually worked with Dan Fenari, who used to be the chief scientist for Deep Submergence at Woods Hole. He's now director of their Deep Ocean Exploration Institute, and it's been put out multiple times in the past, or variations of these other examples of these little planets have been put out on past Alvin cruises. So we're just cycling between cameras now. And we're going to put this down just over on the right hand side of that square rock. It's a square rock's a natural feature that we've been able to recognize over and over again on multiple visits. We're going to aim to put this little marble out over on the right hand side, and then once that's done for this morning, then we will switch to our temperature probe, and you can see the fluid flow down to the bottom left hand side of the square rock. But first, let's see how this pans out. So I think we're probably going to switch back camera views, because Casey needs to move us a little bit closer to be able to reach where we want to get to.
Marble deployment.
So we're just lifting up the submarine and just doing one more of these little rabbit hops to move ourselves just a little bit closer.
Eager in marble anticipation.
So we've repositioned ourselves again, and we're ready to go again with our planned deployment. Do we want to zoom back in with the other camera again? Or are you happy with that field of view?
Entirely sure I can actually reach this. I think we're sitting up on something. See how far down the arm is already?
Mm-hmm. Can you reach the top of that rock that's to the right? You might just roll down into that crevice. Okay.
Yeah, 'cause there's a lot of other rocks. How do I do this?
If I see what the temperature probe, I'll come back up.
Well, you know, if you put it somewhere that's in the field of view forward of the rock, of that square rock, the fluid flow, that could work as well. Let's see how you do with... It's pretty lumpy terrain down there, isn't it? The good news is if we set up somewhere where you can put the marble down, then should be good for being able to do the temperature probe afterwards as well. Okay.
That rock right there on the right.
So while we were setting up for one project, what we actually ended up doing is something else we wanted to do while we're in this location. You can see in their field of view down to the right are the bone bags that Cindy put out in spring of 2012. So we're just going to set up and see whether we can't take a look at them. They were put out with the ROV Jason in 2012. They were revisited, like we revisited some other experiments yesterday, in the spring of 2013 with the ROV Isis, with John Copley from the National Oceanography Centre in Southampton was chief scientist for that program. We have the opportunity to look at them now and see how things are changing with time on this visit, and then Cindy's going to be back again later in this summer as chief scientist aboard the EV Nautilus, at which point she's planning to actually retrieve the systems and see what animals have shown up and what's begun to grow there.
So Cindy Vandover here from Duke University. And as Chris was saying, we put out these bone bags on an earlier cruise. The bones were fetched from a butcher. They're cow bones fetched from a butcher by Bernie Ball in my research laboratory. And what we're after on these bones are a worm called Osidax, which are bone-eating worms. They were discovered by Bob Bryanhook and described by Bryanhook and Greg Rouse. Bryanhook said the Monterey Bay Aquarium Research Institute, and Greg Rouse is now at the Scripps Oceanographic Institution. And these worms are really quite remarkable. They're related to the tube worms you see on the sea floor. They are a species of tube worm, Sebaglinid, and they have no digestive system. They have bacteria inside of them that are somehow using food, chemicals from the decaying matter in the whale bones or in the cow bones. So the cow bones are a kind of a mimic, a substitute for whale bones. So natural bones on the seafloor are large. When whales die, they drop to the seabed, and as the meat is taken away by scavengers, the bones are left behind, and the bones are lipid rich. And then a whole community of chemosynthetic organisms develop around the whale bones. So this cow bone fall is just a mimic for that. We've put these bones at Barbados seep sites, at these Cayman sites at Blake Ridge, and also we'll put them in the Gulf of Mexico. So we are following along with many others who are putting bones, distributing bones throughout the oceans, trying to understand where these Osidax tube worms live, where the different species occur, how many there are, and how they connect, how populations connect from one place to another. So when we pick these back up in August, we will be looking very closely at the bones and dissecting out any of the Osidax worms. We'll then extract DNA from them and compare them to the DNA from others related species. See if we have anything new or not. So it's a way of looking at the diversity of these worms on the seafloor. On the rocks in front of you, you see not just the tube worms, but also little white specks. Those are a new species newly described species of gastropod. Now we have a close-up of the bones, and when the bones were first deployed, the shrimp, the green shrimp, Lebius varantova, immediately were attracted to the bones, to the smell of, to the odor of the meat. They still had just a fine bit of flesh still on them, but now they seem to be pretty ignored by the carnivorous shrimp. And it's hard to see if there were live osedacs in abundance on those bones, we would see a lot of red plumes, and it's just hard to make out anything in this video. You see to the left tube worms. We should have 2 species in this area. And I don't see a good example of one with a plume exposed to point it out to you. When the plume is exposed, it's blood red, and that blood is a hemoglobin pigment, and it's a special hemoglobin in that it both picks up oxygen as well as sulfide, so it's a molecule adapted to extracting sulfide and oxygen at the same time without competition. No, I'd leave it. You want to move it? Is that what you said? Yeah, I think the squat lobster's underneath the bag. So this species of tube worm, these 2 species of tube worms are known from the Gulf of Mexico. Chuck Fisher at Penn State and his students were the first to show that, at least in the Gulf of Mexico, these animals can be quite old, that they grow very slowly, and they might grow fast at certain stages, but they can live to very long, very old age, sometimes centuries old. We do not know growth rates here in these vent systems. We tend to think of animals living at hot springs having really rapid growth rates, rapid reproduction. So we don't know if this species responds differently in this kind of vent environment. I know that at least one of my lab group back at the Duke Marine Lab is listening in, so hello to Bernie Ball. Is that a mussel down to the left? Don't you see that little? I don't think it is. In the Gulf of Mexico, these tube worms have very long roots that go down into the sediment quite deep, and when they form those clusters, they form root balls, kind of manner of speaking. And the tubes are very thin, and sulfide passes through the tube fairly easily. Again, that's work that Chuck Fisher's lab has done, showing that may be one route of nutrition to the tube worm. Through the sediment pore waters into the tube. We don't know if that's the case here. These tubes don't penetrate very far into the sediment, and in fact it's not really sediment, it's rubble, that talc rubble. When we pull them out, they don't have very long root complexes at all. They're just a couple of centimeters into the substratum. We think those snails that you see, the little white snails, are grazers, grazing on bacterial mat. So for scale, these tube worms are pencil diameter, and well, maybe about as long as a pencil. Some of them are longer. And we had a search image when we were here with Jason. We learned which tube was which. So I think in the lower right you see a fairly straight tube, out of focus, but relatively straight compared to some of the others. So that's one species, and then the coiled up tubes, the twisty tubes, are the second species.
There's not enough room for the whole basket to lay down.
So our team at the Duke Marine Lab is interested in these in tube worms. We sampled them with Jason as part of our SEEP program. And although this is a hydrothermal vent, since it shares the same species, we are comparing the genetic composition of these animals with tube worms from Barbados and the Gulf of Mexico. We're working, this is an NSF, National Science Foundation funded project. We're working with colleagues at North Carolina State University who are studying the physical oceanography in the region. So from Barbados up the Gulf of Mexico and on up to the Blake Ridge environment, SEEP environment off of North Carolina. And we are looking at how these populations are connected genetically and also through the physical oceanography. So we get what we call a seascape view of the system. And we will also have included in our team Craig Young and Svetlana Moskova from University of Oregon. And Craig especially is the world's finest, one of the world's finest deep sea larval biology experts. Certainly in the U.S., he's the one we go to. He's studying where the larvae are in the water column, and when they're in the water column. And then we combine all this information, the genetics, the physical oceanography, where the currents are going, and the larval behaviors, and can really begin to understand how the populations are connected, and some of the isolating factors as well.
So we're making a quite change of plan here, because the everything we're looking at here in the rocky area, the boulders are so rocky at this site that they're actually below the bottom of our basket. So Casey doesn't quite have the length of reach on that magic wand to actually put the little marble out on the seafloor. So we're going to put it back in our basket as delicately as we can, and we will plan to put that back out later, probably in a different location. And what we're going to do instead is pick up the probe for the super sampler, which is actually, as you can see in our field of view right now, a much longer instrument on a longer T-handle. And what we'll do with that is use that to probe some of these areas. We can see these little crevices all across the center of the field of view and over to the left of the bone bags. You can see wherever the cracks have that light white coloration, that's either mineralogical or it may actually be microbial filament. And so we're going to check out what the temperatures are there. If they're sufficiently hot, if they're about 50 degrees or more, then Julie's going to be interested in sampling there for microbial activity. And if it gets to 50 degrees and above, Jeff, Jill and Sean are also going to be interested to take their fluid samples as well. Excellent. So we're just getting ready to target some sites. Let's not stick the probe in until we've had the chance to actually see where the fluid flow is.
Yeah, I'm just saying if I could get there. Yeah, I don't think I can. I mean, that's as far down and out as I can go.
Okay, so then that other site's probably even further away, right?
Yeah.
That's the bone bag.
I don't know, up that way.
I think over to the left, yeah. That doesn't look... They don't look especially active in terms of fluid flow compared to what we were looking at before.
And that's where
So now we're just busy repositioning ourselves so we can get back to that square rock we were looking at earlier, which seemed to have the best active fluid flow. And then we're going to put our temperature probe in that. We've done our bone bag imaging and ready to move on to the next phase of what we're doing here. So we can see this whole area has these little cracks with white staining around them, indicative of fluid flow, tube worms sticking out from in amongst the boulders all over the place. But it's such rugged terrain, and the problem with having a flat-bottomed ROV is it's a bit of a challenge to find a place where we can actually settle down and actually do the sampling we want to do, rather bumping around on the seabed as we get ourselves straightened out.
considering the larval biology of these animals and suggests that they can't make that transit around from the Gulf of Mexico around the Florida Peninsula up to the seeps on the continental margin of the U.S. So it's a hypothesis to be tested as we discover more and more seeps off of the coast. So you may be wondering what the difference is between a seep and a vent. Sometimes those terms are used interchangeably. The way I think of it is a hydrothermal vent is something associated with a hot rock somehow, so a volcanic system like the Cayman Rise. Whereas the seeps I talk about in Barbados and Gulf of Mexico, there's no volcanism associated with those. That's in that case, the sulfide is being squeezed from pore waters that are relatively cold, that are cold or just barely above ambient.
Sorry, keyboard's screwed up.
Oh, and my. Sorry. Badly. So seep versus vent. What's interesting about the Cayman site is that it has these tube worms that we've so far only associated with seeps. So we don't know where they originate, whether they adapted first to the vent environment or the seep environment. My bet is on the seep environment. They've been able to opportunistically colonize this environment at the Cayman vents.
We're trying to come in straight over the hill.
Right. Along the spine.
So we are using Nereus to look for warm water. Once we get down on these kinds of terrains, it's really hard to navigate to a specific site. Okay.
So we're having all kinds of fun just at the moment trying to relocate where we saw the fluid flow earlier and trying to get back to the same location. So we have a little area coming into view just now in the centre of our basket. You see some little cobbles down at the bottom of a gully, white standing around the edge. We have shrimp swimming around. And I think we may be in luck. We'll wait and see. So we're just going to have a look on the zoom camera. Waiting for it to power up. And we can tell at a glance that there's nothing vigorous enough in flow there. That's certainly not matching what we were seeing earlier today. So let's switch back out again with cameras. And we'll move on again until we do find something we're excited about. we're facing the right direction. Yep. And we did go over a lot of white-looking rocks just now that would normally have been the cue that I would have used to zoom in on something. So that's us pacing the same direction we were when we first set up trying to do that, putting the marble out. We have our marker off to the left of us, should be come into view on the left as we reverse back this way. I think if we're going down at the same time, then that's a different way of pulling tension on the dispenser. Do you remember, Cindy, whether we saw any little white gastropods when we were looking at it? Oh yeah.
What's that?
That was going to be my leading question is, do you remember seeing gastropods? I thought I remembered seeing little white gastropods when we were there, and that rock there, I believe so. It's okay, we have something of an oil tanker, we don't get to stop as soon as we see it.
Good morning. Good morning, world. We are diving at the Damm hydrothermal vent field. We're at 2372 meters beneath the surface. Got in the water around 4 a.m. this morning. We are in an area of diffuse venting, low temperature venting. All right, what we're doing is looking for shimmery flow in this area, which has proved challenging in the past and is proving challenging this morning. This is an area with tube worms and squat lobsters. Yep, that's it. And snails. I have Cindy Vandover over my shoulder, our biologist on board. There are 2 types of tube worms here, and they are not related to the large Riftia type tube worms seen in the Pacific Ocean. They're related to tube worms seen in methane seeps and Gulf of Mexico and Barbados. My cue cards. So what we want to try to do is get a sample in this diffuse flow, because it's unusual to see seep-like worms in a hydrothermal vent setting. At the summit of this large mound, there's 225 degrees Celsius fluid, but down here at the base, it's very diffuse and leaky, and we have these seep-like worms. So, okay, great. So Cindy's lab is working on the genetics of the tube worms. My lab's been looking at the symbionts inside the tube worms that makes living at a vent possible for these worms. So right now we're picking up the temperature probe so that we can try to sniff it out, see what the temperature is. What time is it? 8:30 in the morning. We're on Eastern Standard Time here on the ship, which is very convenient. Hey, we're back where we were. So we're trying to get our sampler intake out of the milk crate. Milk crate is a common tool of deep sea oceanography. It's easy to attach to an ROV basket, and you can kind of just drop whatever stuff you need in there. We also have in there a net in case we find any mussels. There we go. We don't want to sample the net. We want to sample the fluids. Pilots always try to grab at the T. Most of our equipment has a T handle on it, which is easiest for the grip of the robotic arm to pick up. So the reason Nereus only has one arm is because Nereus is designed to go to the deepest part of the world's oceans, and every pound is accounted for. The arm weighs about 110 pounds, and it was manufactured by Craft Robotics and designed to be as light as possible, but still very agile. Has 7 functions. You can see it has a wrist, has an elbow, has a shoulder, and the claw itself can spin. I'm sure there are other functions that add 7, but I don't know what they are. Nancy, Casey just named them all. This has force feedback too? Okay, that Jason ones do. They never use them though. Yeah, it probably helps with coring.
I core by smell.
So we're gonna see if we can get a temperature. You can't see the video where we're trying to put it really well right now. Or just now that we have the temperature probe in our hand, we're gonna reposition a little bit. And again, this is sort of a rubbly area at the south end of Von Damm. Later on, we'll be proceeding sort of up the mound, and you'll be able to see more vigorous venting. But unlike where we've been diving the last couple days, Picard, there's no black smoke here. The highest temperature fluid we see is 226 degrees Celsius, whereas we see 398 at Picard. And these two sites are only 20 kilometers apart, but they have 2,700 meters difference between them. And they're very different just visually. Chemically, they're different. But just by looking, I think you can tell it's a very different place than where we've been diving previously. There's very little metals in the fluids here. The other site had tons of iron. And probably some other metals. And the fluids here have very little iron in them, which is why we don't see any red staining like we did at the other site. You can see a squat lobster up in the top there. You can't reach it? Inches away. We don't know the temperature. Well, we're not in it yet. What? No. We're still pretty. So here we are trying to poke our sensor in there, but we're just about just an inch or so away. They can see the control van?
Occasionally takes a picture with that camera. Once every 10 minutes.
We're only exhausted every 10 minutes.
So we're going to try to slightly reposition the vehicle, see if we can just get a tiny bit closer so that temperature probe and sampling intake can reach into the vent that we're interested in getting a temperature on. These big rocks and kind of get in the way of doing science. I don't think I'm allowed to say that word on air. So, for those of you just getting into the office and looking for something to distract you, we are here at the Von Damm hydrothermal field, named after the vent chemist Karen Von Damm, who did some groundbreaking studies in vent geochemistry. This is a really interesting site. We're at the base of the mound, and all those curly things you can see, those are tube worms. Their guts are packed with symbiotic bacteria that are extracting chemical energy from the vent fluids to provide carbon to their animal host in a chemosynthetic relationship. And also the tiny little white dots you see are gastropods, little snails. See a few shrimp around here, and every once in a while we also see a squat lobster or two. Squat lobster actually a crab? What? It's a crab?
No, it's a spot.
Oh, it's a lobster?
Spot lobster.
Okay.
So crabs are brachioids.
Uh-huh.
And spot lobsters are annelids.
Oh, okay.
Decapod
Okay.
crustaceans.
I think squat lobsters are cute.
They are.
I think they are. So we're just trying to reposition here again. I think this big rock might be part of our problem. And then there's another big rock. Can you go up slope at all?
Yeah, we were set up there before, sort of, see all these big rocks?
Mm-hmm.
Oh,
And the whale bones.
Like the middle of
If
trying
you
to sample from the little Stonehenge.
If you think the bones are in the way, and they're not whale bones, they're cow bones.
Cow bones.
But if you think the bones are in the way, you can kind of gently move them over to a different position.
So
Fine by me.
last time we were out here, Cindy deployed a bag of cow bones on the seafloor. A couple bags.
One's wood.
One has wood in it and one has cow bones. And they're interested in seeing colonizers. Those are both really organic, rich energy sources for various invertebrates. And some very interesting species have been found on wood and bones in the deep ocean, and they're related to the types of organisms we see at hydrothermal vents. There's actually, so every time a whale dies, it has to go somewhere and it sinks to the bottom of the ocean, and there's a whole unique group of organisms that colonize and extract energy from it. Because in the deep sea, except at places like hydrothermal vents, organic carbon's a little bit hard to come by. Just saw our marker up there in the upper left.
Where's your map of the seafloor? Is that on there? I've got all the relationships like that.
Here it is. The arm just... I think the rock is actually...
So his rocks at 18. So I see it's 18. Just there. And then so I see it as
I think the rock's more like here, right?
rock.
Here. Cindy and I are just trying to get our bearings a little bit better here.
And this heading is.
We're on the right heading now. From
Headings
the
are
left
headings.
line.
Yeah.
068.
And it's just up from that ruler.
So does that look like whale bones, ruler rock, rocker?
Yes, that looks correct. What? Rock and roll site, yes. These large boulders makes setting down the vehicle really difficult. And then we're also on a slope. So some tough terrain. Pardon?
So top of the rock. And
So what are you trying to do right now, Casey?
I'm just seeing if I hit it, if it moves. But I can't.
I don't think that's good for the vehicle.
Might be better off moving the whale bones if that would help.
Well, I was gonna see if it moves, then I can move over and try to roll it down.
What about, can you sit in front of those whale bones? Is there enough space?
The back of the basket, I'll be sitting on it.
It's okay to sit on them, but it doesn't help you get into where you need to go.
Yeah.
No, I've suggested these robots to move them over.
I mean, I don't know if that's what I'm hitting from that angle, but that would avoid rock A and B.
We can put them over next to Rock A.
We're going to move our bag of cow bones over a couple meters to see if that clears some more space to set the vehicle down at this area. Again, these were cow bones deployed by Cindy in January of 2012, and they're interested in the unique invertebrate colonizers. Invertebrate, I guess, but mostly invertebrate. Okay.
We'll have to,
It looks like there's a crab inside there, or a squat lobster inside the bag.
which would be very interesting.
But they're probably going to eat all the worms if they get in there.
Yeah, but how did it get in there? Did we rip the bag?
I don't know.
Or did it come in as a baby?
Right, a larvae.
That'd be
Yeah,
cool.
that would be cool. Spend his whole life on a cow bone, thinking
All
this
the
is
other bags
awesome.
that we're trying to capture.
Life's not hard in the deep sea. So we're going to stow the sampler to give ourselves a chance to move the whale bones. Sorry, cow bones. Cow bones.
Before he moves, could he just zoom in and kind of
Can you zoom?
clear water?
Wait, hold on. Oh, okay.
Just it's a nice,
Hold on.
No, go ahead. We're
Where would you like me to put them?
wherever you can reach with them, it doesn't matter too much.
Isn't-
They
is-
don't need warm water or anything. It's looking just for what's on the boat. So if you go off to the left, off to the right, well, that's going to be out of view, right?
Do you wanna
Put them near the other rock.
yeah, near the big rock.
That's the rock question that we're sitting on. Oh, so that's the other one. Yeah. I've marked it.
There is a squat lobster.
He crawled into the slots. That would be great.
All right, we're going
Have
to
the time to do that.
zoom. Well, that was on the dive plan, right? To take a look at your whale bones. Sorry, cow bones. Cow bones as a proxy for whale bones. It's hard to come by whale bones. Cow bones are easier. Yeah, there's a squat lobster inside the bag.
I don't think so. I think he slipped in under.
So we're going to just zoom in and get some high-def video of the cow bones and the interesting colonizers. 2. Yeah, Cindy has it. This is number 2. So why are we putting cow bones at a hydrothermal vent? It's a good question. But, oh look, yeah, you can see the lobster in there. So in the deep sea, most of the deep sea is, but especially heterotrophic, there's a whale dive away from shore